Well, let's get to work. We have a lot to cover. Let me begin, as usual, with a few words about the last lecture to bring you up to speed and establish some continuity. In the last lecture, we talked about something called the Hardy-Weinberg law or theorem. It's a very simple calculation. The first step was to calculate the frequency of a gene in a population. After calculating its frequency, we equate the frequency to the probability of drawing that gene from the population. And then we assume that we have to draw two copies to form a diploid individual. And we assume that that draw is done independently so that we can multiply the probabilities, and that gives us the genotype frequency distribution. A question?
Can you turn up your mic a little bit?
Let's see if I can get this so it. Let me try it on the other side. Well, let me go on. So that's really a very, very simple calculation. It's very obvious. It's nothing more than the simplest of elementary algebra. But the great thing is that it gives you the genotype frequency distribution after a generation of random mating. And then you can use that as a way of testing different hypotheses, and we'll see some examples of that in this lecture. The really important fact, though, is that the gene frequencies do not change under successive generations of random mating alone. So random mating has no effect on gene frequencies. Therefore, it doesn't cause any kind of evolutionary change. The other really important fact is that it implies the conservation of genetic variance. Okay? Remember, I've emphasized that you have to have genetic variance, that is, differences among individuals that are heritable, in order for natural selection to have any effect. You can have selection, but if it isn't causing genetic change, it isn't going to lead to adaptation in future generations. Particulate inheritance does not change genetic variance. It conserves genetic variance. And we mentioned the fact that one of the great misconceptions of Darwin and of almost all of the other thinkers of his generation was the notion that inheritance was a blending system, which would not conserve genetic variance. So those are the really crucial facts that we talked about in the last lecture. Then we went on to say that there are 4 evolutionary forces that we're going to consider, and we're going to focus on those mostly in this lecture. And they are selection, natural selection, which is the most important one, which is the fact or the operation or the mechanism that leads to adaptive change over time. There is genetic random drift, which is purely random, does not lead to adaptive change. There is migration of genes among populations. And finally, there is mutation, which is the ultimate source of all genetic variation. It all has to arise as a mutation at some point in the past. So we're going to talk about these forces today, but I want to go back to the slide that I ended the last lecture on, just because I thought we went over it too quickly. We were looking at the frequency of genotypes at the imian locus. It's a blood group gene. And we wanted to ask, is mating at random with respect to the imian locus? And we actually have real empirical data from a number of different human populations on the frequency of genotypes within each of those populations for imian. And at the bottom, and this is taken directly from your textbook, is the ANU in Japan. And as an exercise, you're asked to calculate what the expected genotype frequencies ought to be given the observed data. Well, how do you do that? Well, I've used F to stand for frequency of whatever's inside the parenthesis. So you want to calculate the frequency of the capital M allele. How do we do that? Well, it's all of the MM homozygous class. That frequency is 0.179 plus half of the heterozygous class. The heterozygous class is 0.502, and half of that is actually should be 0.41, right? Should be 0.26. I don't know why we go, well, interesting. Okay, so there's an error. 2 into 50 is 25, so this is 0.255 is half of the heterozygous class frequency, and if we add those two up, we get 0.42, okay? So the frequency then of the capital M allele is 0.42. The probability of drawing two independently to form a new homozygote is 0.42 squared, which is actually 0.176. And now we put that in the expected box there, and we can compare it to what was observed, 0.179 versus 0.176. Well, that looks pretty close. Good enough for government work, right? It fits very well. Now we can continue this kind of calculation and fill in the other two open boxes. The heterozygous class is twice the gene frequency for capital M times the gene frequency for capital N, and that's 0.487 as compared to 0.502. Again, very close. Now, it happens in statistics. There are ways to actually ask objectively, are these so close that they're the same within the context of a random draw from a population of this size, all right? Some of you will have had elementary statistics, and you'll know how to do that test. It's called a goodness of fit test, but I will tell you that the fit is very good, and it is highly likely that it is so good that we can easily accept the hypothesis of random mating with respect to the MN blood group locus. Now, is that a surprise? No, it isn't, because people don't ask their prospective mates what their genotype is at the MN blood group locus. It's just not it's not a big concern. So the chances are that they're random choices. Now let's look at a more interesting case where we try and look at calculating deviations from this Hardy-Weinberg law. And this is a case of a chemokine gene called CCR5. It encodes a protein that actually sits on the leukocyte white blood cell surface. And it happens that that protein is the route by which the HIV virus enters the cell, where it then replicates for some period of time beneath the surveillance of the immune system before AIDS is developed as a full-blown disease. So this is a very important protein. It is the route by which the HIV virus attacks the immune cells. And there happens to be a mutation in human populations in CCR5, which is a 32 base pair deletion in the gene, in the protein, changes its length and structure. And we're going to call that the del32 mutation. And we're going to ask some questions about this. We're going to use the Hardy-Weinberg rule as a way of asking questions. We're going to ask, do HIV positive individuals with the del32 mutation show a reduced progression to AIDS? We're going to ask, is there a difference between del32 homozygotes versus heterozygotes in this regard, in progression of the disease? And finally, we're going to ask, what do we know about the origins and distribution of this del32 mutation? So the first question is exemplified here. We use the Hardy-Weinberg law to calculate the genotype frequencies among individuals right after they have become HIV positive, within 1 month of becoming HIV positive, for the 3 genotypes: the normal, which we're designating with a plus, the heterozygote, +/32 to stand for the del32 mutation, and the mutant homozygote, which we're denoting by 32/32. Well, after the frequency of the normal allele happens to be about 0.9 overall in the human population. If it's 0.9, what is the frequency that we would calculate for the normal homozygote? 0.9 squared, which is 0.81. And in fact, that's what we see shortly after people have become HIV positive. Their frequency for the normal allele, nor the genotype with the normal homozygote, is just what you would predict based on random mating. Likewise for the heterozygote and the other homozygote, the del32 homozygote is very rare because the gene frequency is only 0.1, so the homozygous genotype frequency is 0.1 squared under the Hardy-Weinberg law, and that's exactly what we observe. But people followed these genotypes in a cohort of individuals who had been diagnosed as HIV positive, and that cohort was followed over a decade. Of course, during the decade, many of the people in the cohort died of AIDS. But after 1 year, you see that the frequency of the normal homozygote has declined. It's only 0.75 now. It's declined because of mortality. After 5 years, it's declined to 0.2 because of mortality. And the frequency of the del32 genotype has gone up so that after 10 years, it accounts for 75% of the individuals in the cohort that was followed over this decade. Now, what does that tell you? It tells you that del32 confers strong resistance to AIDS. Why is that? Well, we know it is because the 32 base pair deletion mutation affects the CCR5 protein in a way that apparently prevents the HIV virus from entering the cell. We also can conclude from this experiment and the calculations that being a heterozygote confers some resistance as well, but not perfect resistance. So the heterozygotes have an advantage. Being heterozygous for the del32 mutation gives you an advantage. So this is a good example of the use of the Hardy-Weinberg law to make important deductions about a public health problem. Well, one can go further. We can actually look at a global population of people and calculate the frequency based on geographic origins of the del32 mutation. And we find that people from Africa and from Southern Europe simply do not have the del32 mutation. Its highest frequencies are in Northern Europe. And then there's as you move from Northern Europe down into Southern Europe, this is a map of Europe. You can see Italy sticking out, North Africa, the Middle East, on towards India and Asia. The dark red represents a frequency of about 0.18, down to the very light yellow representing a frequency of about 0.01 for the del32 mutation. Now, what does this lead you to infer about the origin, the geographic origin of the del32 mutation? Well, probably its geographic origin was in the part of the world where its frequency is highest, in Northern Europe. So for some reason, probably several thousand years ago, this mutation arose in Northern Europe, and it has been favored. Not because of HIV, because HIV is a modern disease of Europeans, but because it apparently conferred resistance perhaps to other kinds of infections, and we don't know for sure what they were. One hypothesis is smallpox. Smallpox, by the way, is another example of a zoonotic disease acquired from an animal associate of humans after the invention of agriculture. So maybe, we don't know this to be the case, it's simply a hypothesis, but maybe the del32 mutation has been under selection in the human population for a long time, possibly associated with another disease like smallpox. And we can infer, based on the geographic distribution, that the mutation probably arose in Northern Europe, where it is currently the most frequent. What can we predict about the ultimate future of the del32 mutation if AIDS continues to be a serious health problem for humans? Is it going to go down in frequency or up in frequency? Don't all answer at once. It's going to go up in frequency. If it continues to favor people's survival in the presence of the disease, it's going to be selected upwards. That's natural selection in action. What about the heterozygotes? Well, they have some advantage too. So individuals who are heterozygous are going to still have an advantage in survival and reproduction relative to homozygotes for the normal CCR5 protein. Okay, so that's an example of a really good contemporary example of natural selection. Now, we're going to talk a bit about patterns of natural selection. The first thing we're going to talk about is heterozygous advantage. We just saw an example where the heterozygote is favored, but not as strongly as the del32 homozygote. But there are other cases where the heterozygote has actually got an advantage over both homozygotes. That happens to be the situation with sickle cell anemia in areas of Africa where malaria is endemic, because the heterozygote actually has the best of both worlds. It has some protection from malaria. It doesn't have the severe anemia. It has mild anemia, but not the severe anemia suffered by the mutant homozygote in the case of sickle cell anemia. So we're going to talk a bit about heterozygous advantage. Now, what do you think the consequences of selecting for heterozygotes would be on the change in gene frequencies? Well, heterozygotes have both genes. So if we're favoring heterozygotes, we're going to keep both genes in the population, aren't we? Okay, so that's heterozygous advantage, and there's a table in your text with a different example, which is kind of a nice one. This is HLA, the human leukocyte antigen, HLA, major immune system, gene system in the human genome. It's the thing you need to type if you're going to have a transplant, a tissue transplant, because the HIV antigen will lead to the rejection of non-matches. Now, it turns out that HLA is a complicated gene. It's a set of a number of genes with many, many mutant alleles. So there's a large number of alleles. But we can still apply our Hardy-Weinberg law to ask whether heterozygotes are favored at HLA. But we're going to do it in an easy way. We're just going to sum up all of the homozygous classes and all of the heterozygous classes and represent those as just two different classes. The sum of all the homozygotes, the sum of all the heterozygotes. And this happens to be data that was published 20 years ago in the American Journal of Human Genetics. And it's actually a Native American population called the Havasupai. And what is done is to calculate the the observed number, to estimate the observed number of each class of genotypes from a real sample for two different HLA loci, A and B, and then compare this to the Hardy-Weinberg expectations. And when you do that, you see that there's a deficit of homozygotes and an excess of heterozygotes observed compared to what one would expect under the random mating assumption. Okay? And one can do a statistical test, and if we do it, turns out that it's very unlikely to have drawn an outcome like this randomly. So we can conclude that there's something going on which is favoring heterozygotes at the HLA locus. And your book talks about two possibilities. One kind of interesting possibility is maybe people mate based on their HLA genotype. Okay? That's called negative assortative mating. In this case, unlike individuals choose to mate. There's some evidence to actually support that because it turns out that human odor is a little different between different HLA genotypes, and people tend to prefer those who smell differently from themselves. So maybe there's negative assortative mating. But the other possibility, which is also very likely, is that being heterozygous for these very important loci gives you an advantage in disease resistance. Okay? So this is heterozygous advantage. It really does happen in the real world. Now I'm going to go fairly quickly through other patterns of selection. They're all in your book and they're all obvious. One is directional selection. Well, we saw that in our example of HIV and CCR5. It's simply where there's selection for one end of the distribution of phenotypes. And it might be selection for faster runners on the savanna, for example, if you're threatened by large mammal predators. But whatever it is, if it's heritable, directional selection eventually leads to the fixation of genes that favor that phenotype. So here's directional selection as illustrated in a frequency distribution. In this case, the right end of the distribution are the favored individuals. After selection, the distribution is shifted to the right, and that goes on generation after generation until you end up with a permanent shift in the phenotype to the right. Another example we saw a couple lectures ago was the artificial selection for high protein in corn seeds. So that's directional selection. There's an example discussed in your book of directional selection operating on cliff swallows in the natural world. In this case, a population of cliff swallows had been studied for some time. There was a period of extreme cold and heavy rain, a week or two, long enough period of time that it really stressed the population. Many individuals died of starvation because the cliff swallows depend on being able to catch small insects to eat, like mosquitoes. But if under these weather conditions, the small insects weren't out, the food source was gone, the cliff swallow population was stressed, and the result was that the original population, which is shown in this light green histogram, had very high mortality. The survivors were then later measured, and their mean size, body size, had shifted to the right. Why? Well, your book speculates that the reason is that the large cliff swallows were fatter and had more energy, and therefore were in a better position to survive this period of starvation or stress. So there was directional selection for larger body size. But under normal times, large body size for cliff swallows means that they're not as agile flyers, and therefore not as effective in capturing insects. So under normal circumstances, selection might reverse this trend observed in this one episode. And so that's directional selection. Another kind of selection is stabilizing selection. That's just what the word implies. Here, both extremes of the distribution are removed, okay? So it's the intermediate traits which reproduce more than the ones on either extreme. And here's an example of the actual operation of stabilizing selection. You have a normal distribution. The two extremes are both selected against reducing the variation, but the mean of the distribution remains where it was. It's just that the extreme phenotypes are removed. There's a really nice example of this, and that's birth weight in human babies. It turns out that mortality for human babies is high at low birth weight, but also it's higher at high birth weight. And these are some actual data which were collected in Great Britain that illustrate this fact. The x-axis gives a birth weight in pounds, and the y-axis gives a percentage of mortality in the population on the right side. And the blue curve just shows the curve of mortality as plotted against birth weight. And you can see that mortality is minimized at about 7.5 pounds. So this is a good example of stabilizing selection in humans for birth weight. Another kind of selection is disruptive selection. Now, what would disruptive selection would be? Well, that's just the complement of stabilizing selection. Here, for some reason, the intermediate type is not favored relative to the two extremes. So you're taking out the middle of the distribution, where the people in the mean and the middle part of the distribution have a lower probability of survival and reproduction relative to the two extremes. Disruptive selection, of course, is going to maintain genetic variation because it's saving the extremes. And here's an example from your book of the operation of disruptive selection. Each of the two extremes are high fitness, the mean is low fitness, and you end up with this kind of bimodal distribution after selection. That's disruptive selection. And there's an example of it in your book. It has to do with the beak size of black-bellied seed crackers. And it turns out that there's just two sources of food for black-bellied seed crackers. They are small seeds or large seeds, so they have this discretely different resources available to them. The small beaked ones are very efficient at breaking open and eating the small seeds, but not the large seeds. And conversely, the large beaked nutcrackers are more effective at breaking open the large seed and eating from them. So selection is on the two extremes. And under some circumstances, and we'll talk about an example in another lecture, this kind of disruptive selection might even lead, if it goes on for thousands of generations, to the emergence of two different species. Okay, that's the patterns of natural selection. Okay? That just a classification of the different ways that natural selection can affect the distribution of variation in a population. Now let's turn to genetic drift. Genetic drift is just the random sampling of genes to form the next generation. Remember, we talked about this abstract idea of a gene pool. So suppose that we have this abstract gene pool, and we sample from it 100 genes to form a new population of 50 individuals. Now, the gene pool is thought of as being almost infinite. Why would we think of that? Well, because the number of gametes that we can produce relative to the number of offspring we produce is vastly larger. So there's a large number of gametes but a relatively small number that will actually be sampled to form the next generation. So it's just a random sampling. And those random draws lead to small statistical fluctuations in the gene frequencies from generation to generation. An important point is that genetic drift is random with respect to fitness. It is not an adaptive kind of genetic change. It is random with respect to adaptation. Okay? So that's an important question, the sort of thing you'd be easy to ask an exam question on. The allele frequencies under drift, the allele frequency changes are not adaptive. Now let's think about a kind of an interesting case. You all know about the HMS Bounty. The HMS Bounty was a British ship which was actually sent to Polynesia from Britain with the mission of collecting breadfruit plants, which they hoped could be used as a source of food. And the captain was a guy named Captain Bligh. His name has become sort of synonymous with being hard and mean. And he drove his crew hard. They arrived in Tahiti and actually spent about 5 months there collecting breadfruit and growing them to the point where they could put them on the ship to take them back to England. But on the way back, the crew mutinied because Captain Bligh was so hard to work for. And the leaders of the mutiny were his first mate, Fletcher Christian, and about 18 other sailors. There were actually 42 sailors on the ship. They mutinied, and the mutineers put Captain Bligh and 22 of the other sailors in an open, large rowboat on the open sea in the Pacific, and they sailed away. And Captain Bligh managed to actually navigate over 3,000 miles to New Guinea and made his way back to England. The mutineers returned to Tahiti, but they knew that they'd be caught sooner or later and hung. So they, by now, just about 12 of the mutineers, plus 18 Tahitian women, plus 6 Tahitian men, left on the ship and went to an island called Pitcairn Island, which was incorrectly mapped on the charts anyway, and they were unlikely to be found there. And they established a kind of colony, and it's kind of an interesting history to it and so forth. But this was a small population of individuals who began on Pitcairn Island. And so we can ask, well, let's just sort of do a thought experiment about how their gene frequencies might change. So let's think of a simple case where we have just 6 couples. There was actually somewhat more. And we'll think about a case where there are actually 3 alleles at a locus, A1, A2, and A3, and they're all color-coded. And look at couple 1. One parent, the father, is a homozygote A3A3, and the mother is a heterozygote A2A3. All right, so the only gene that the father can transmit to its offspring is A3, right? But with probability 1/2, we can draw either A2 or A3 from the mother for transmission to an offspring. Well, it happens that they had 2 children, and by chance, both of them ended up A3A3 homozygotes. The chance of one of those happening is 1/2. The chance that that happened twice is 1/2 squared or 1/4. Not too unlikely. You can simulate this sort of thing by just flipping a coin. Couple 2, heterozygous A2A3, they had 2 children, both were heterozygous A2A3, no change. So we can go through this exercise, and then you can count up, let's say we count up the number of red genes among the father and mother in the first generation, and the number in the second generation. Well, if you count the reds, you see there's 8 among the mothers and fathers, but there's only 7 among the children, just because of the random draw of the genes that were sampled to the next generation. We end up with a change in the frequency of the A2, the red A2 gene, okay? So you can go through that with each of these. That's genetic random drift. It's just a random draw of genes for the next generation. And people have done lots of experiments asking, well, if we do this, if we simulate drift in an experimental situation, will we observe it? The answer is absolutely. It's a mathematical fact. It's a consequence of sampling. But here's an example of an experiment. This is with the fruit fly Drosophila, and there's a gene that determines whether the bristles on the back are forked or normal, straight. It's one gene, and so what was done was to start up a series of replicate populations. Each replicate had 8 individuals, 4 female, 4 males. There were 96 replicates. In generation 1, the 4 males and 4 females in a replicate are put together in a small tube with media. They're allowed to reproduce. And then for the next generation, 4 males and 4 females are randomly drawn from the progeny to do another generation. And this is done for all 96. Well, after only 16 generations, if you look at the top curve, the one that begins with both alleles present, they all began with equal frequencies of forked and normal, 50-50. By the time 16 generations have passed, 73% of the vials have lost one allele or the other. They've become fixed because of the sampling process. The remaining 23% or so are shown in the red and the blue curves. The red and the blue curves show you the number which are fixed for the forked. That's blue. Red is the number that are fixed for the normal bristle length. So you get something that looks pretty much like what you'd expect under random drift. Now, you might ask, well, you've got a higher number of vials where the forked gene was lost, and maybe there's a slight disadvantage of the forked gene, but that would require some careful statistical testing. You can also do this sort of experiment on a computer. You can do a computer simulation, and this is an example of a couple of computer simulations. It makes an important point. The only reason I'm showing you this slide is to make one more point, and that is the obvious one, that the effect of drift is highly dependent on the sample size. If you have a very small sample, the probability of a large deviation is higher. Okay? Think of it, if you're drawing just 2 genes to transmit from one generation to the next, well, think about this in the case of self-fertilization in the moment. Suppose you have A1 and A2 only, and you're going to draw 2 genes for the next generation. A fourth of the time it's going to be only A1 because it's a A1 with a probability of a half, A1 again with a probability of a half. So then you'd have a complete fixation in one generation if the sample size was only 2. So the smaller the sample size, the bigger the effect. So this is a simulation with a population size of 4, and you can see that within 20 generations, all variation has been lost in this computer simulation. On the other hand, if you take a population size 400, even after 100 generations, and each of these lines represents a different replicate of the same experiment, after 100 generations, none of them have become fixed. Because you're drawing 100 copies in each generation, the probability of an extreme deviation is less. Okay? So the effect of drift is much larger in small populations than in large populations. But drift is important, and one place where it's really important is in conservation biology, where there's a concern about the maintenance of particularly endangered large animal species in zoos and so forth. The population sizes that are maintained are very small, and so there's a real concern about the progressive loss of variation to the point where the species becomes labile to things like disease infections and so forth. And there's special mating schemes that are used to try and minimize the loss of variation under drift in these conservation biology scenarios. There's a couple of other places where genetic drift is especially important. These are, instead of having a continuous sampling process that is the same in every generation, these represent cases where there's a big effect in one generation. One is called a founder effect, okay? We saw an example of a founder effect in Pitcairn Island. We had a small number of individuals that was drawn from the much larger Tahitian and British populations, probably didn't look too much like either parental population just because of the random draw of the individuals. That's a founder effect. It's a sampling phenomenon where there's a sudden shift in gene frequencies simply because you sampled a very small number of individuals to found a new population. The other is a bottleneck, right? So these figures from your book illustrate the two. Here's the founder effect on top. Immigrants establish a new population from a small number of individuals, therefore there's a change just because of the sample in gene frequencies. Bottleneck is a different thing, but very similar. This is where something happened and the population crashed to a small number and then gradually recovered, but because of that crash, for some short period, there was a small number of individuals to transmit their genes to the next generation, and therefore an enhanced drift effect. Now, an example that might be worth thinking about is from about the year 700 until the late 19th century, bubonic plague was an important scourge of humans in different parts of the world. It's caused by a bacterial organism which is carried on the lice of rats. And probably the worst episode of bubonic plague began around 1348. And in France, almost half the population died because of bubonic plague in the years 1348 and 1349. It's a huge collapse in the human population. That's an example of a bottleneck. Suddenly the population goes through a much reduced number. Moreover, it took the human population at that time in the whole globe was about 400 million people. Today it's 7 billion, big difference. It's about 400 million people. It took more than 150 years for the human population to reach that number again. So for some period, the human population was reduced relative to what it had been because of the bubonic plague, and therefore the drift effects were larger during that period. So that's an example of a genetic bottleneck. It turns out that humans went through a bottleneck sometime early in the history of humans, maybe 30 or 40,000 years ago. The human population got reduced to perhaps only about 10,000 individuals. And we still see the genetic signature of that in the amount of variation in humans compared to their population size. Humans are not that variable, surprisingly enough. Compared to many organisms, they're only about 1/10 as variable as maize, as corn, maybe only, I don't know, half or less than half as variable as barley. Many other mammalian species are more variable. Humans went through an important genetic bottleneck early in their history, 30 to 50,000 years ago. That's a bottleneck. All right, now let's talk about gene flow. One of the other evolutionary forces. Gene flow is when you move genes from one population to another. And let me talk about a kind of fun contemporary example. But first, what are the consequences of gene flow? Well, if two previously isolated populations are exchanging genes, they're going to become more similar, aren't they, genetically? Makes them more similar. Now, humans have a great evolutionary history. Modern Homo sapiens emerged, I don't know, maybe 250, 300,000 years ago in southern Africa. And they moved up the Rift Valley of Africa slowly as populations grew. And eventually, about 100,000 years ago, across the Red Sea into the Arabian Peninsula, up into what is now the Middle East, and across, down around the periphery of India by 70,000 years ago, up into China by 70,000 years ago, down into Australia more than 50,000 years ago, into Northern and Western Europe about 40,000 years ago. So that's the migrational history of humans. Now, there was another Homo species there before humans. About 800,000 years ago, two lineages of Homo separated. One became Neanderthal and the other became Homo sapiens sapiens that we belong to. Some authorities think Neanderthals are just a different subspecies of Homo sapiens, but whatever, Neanderthals got into Europe by sometime between 350,000 and 600,000 years ago. So they were there long before modern humans got into Europe and Southeast Asia. Now, only in the last couple of years, because again of the elaboration of high-speed DNA sequencing technologies, we can now actually get fragments of DNA from the fossil bones of Neanderthals, and we can ask, how did the Neanderthal genome look compared to the human genome? And we can sequence them. And just in the last couple of years, we have fairly clear evidence that there was a moderate amount of genetic exchange. Somewhere between 1 and 4% of your genes have a Neanderthal origin. Okay? So, an example of genetic migration. Mutation, the other force that we're going to talk about. Mutation is the source of all genetic variation. Now, a key thing is that mutation is random with respect to fitness. Mutations are not, they do not arise in response to an environmental need. The frequency may be increased by radiation or other environmental perturbations, but they're random with respect to the environment and with respect to fitness. Another key fact about mutation is that most mutations are deleterious. Only a few may confer some adaptive advantage. Why is that? Well, it's sort of like, taking a protein and hitting it with a hammer. When you put in a wrong code, most of the time it's going to make something less effective than it was before. So most mutations are deleterious. Now, the last thing I'm going to say is, how many new mutations do we have each generation? Well, we can kind of calculate that. We've got pretty good estimates of the rate of base pair change per generation. It's about 10 to the minus, the probability of having a base pair substitution in a single replication is about 10 to the minus 8 per site, per base pair. There's 3 times 10 to the 9th base pairs in the human genome. So there's 30 new mutations that would arise in each generation. But only about 10% of genes code for proteins, so roughly 3 of those might fall in protein coding genes. So that's mutation, and we'll pick up here when we start again on Wednesday.